Alright, so this isn't the current version that you have in front of you, but close enough. I just want to bring to your attention that next week you will get your very thank you. You will get your very own present. It's called your unknown organism, right? And it'll be assigned to you. You will get a number, and I know what organism it is. You are tasked with figuring out which one it is. What is its problem? Oh, I unplugged it, remember? For the vortex. There we go. Power is always helpful. So, you'll get your very own organism, and the first thing that you're going to do is you're going to inoculate a whole bunch of differential and selective media tubes and regular food to just grow your organism. One of the first things you're going to be tasked with doing is to record on your unknown report, which I'll give you. It's a report that you fill out as you go and determine all these characteristics about your organism. And then eventually you'll narrow it down to one of the 14, I think, on the list. So one of the first things you'll need to do is describe what your organism looks like growing in the slant culture that I'm going to give you. So you guys have already kind of noticed this kind of stuff, and there's a section in your book that goes over characteristics in broth media and slant media. It's in exercise 2. So if you look at your syllabus, right, it tells you, and I would read over that section in your syllabus, and it actually says that on your sheet to do that. And then the other thing is your homework for next week will not be written on these sheets you picked up from me. You're going to do it on a separate sheet or sheets of paper. You're going to write out all your staining procedures, and this is for you to use in lab next week, because you're going to make all the slides and do all the stains next week of your unknown organism. All right, so that handout you have is mostly just for in class. Your homework, as you'll see highlighted, is to write out or type out all the staining procedures listed there. Okay? Which I think, does it say how to make a bacterial smear? But you'll notice for some of those you have to make a bacterial smear. Did you guys remember from your exam, if I give you a slant culture, what do you need first to add to your slide before you can make that bacterial smear? You need to add a drop of water, right? And then you're going to air dry it and heat fix it before you can, say, gram stain it or endospore stain it or acid fast stain it, right? The only two stains where you don't have to do a heat-fixed bacterial smear is the negative stain and the capsule stain. Because remember those, you put the dye, you add the bacteria, you spread them out, and you just air dry them. With the capsule stain, you're going to air dry and then stain, right? So make sure you write out those procedures, what stains, what times, right? So that you can read it and you can understand it and you can use it as a guide next week. That's your homework, right, for next week. Does that make sense to everybody? Hence why Andrew and I took the time to highlight that section on your homework handout for this week. Okay, so, and I'll post this semester's version on our Canvas site. So that brings us to today's lab. So you've already done, right, measuring the Kirby-Bauer, and we're going to talk about that. And I've demonstrated a dilution and spread plate method of spreading out those dilutions in order to be able to count about how many bacteria we started out with. So antibiotic sensitivity, and there are different tests out there. The particular test and method that we used was the Kirby-Bauer method. And so again, just like with acid fast and endospore staining, it's important to know what method you're using. And that will determine how you perform it and how you interpret your results. So some important terminology that we need to know is antibiotics, right? You hear this term all the time, but what does it mean? It means that that substance, that chemical, is natural. It was actually made by a microorganism. And it will kill or inhibit another microorganism, obviously not itself. So we've already looked at a beautiful mold under the microscope that produces a very common antibiotic that most of us are aware of. What was that mold? Penicillium. And what does it produce? Penicillin. Guess what's growing here? Penicillium. Penicillium. It's producing that antibiotic we know as penicillin. It's diffusing into the agar. So instead of a disc, like what we worked with, the actual organism is sitting there, right? Those discs you worked with have different antimicrobials and pigmented into them, maybe even an antibiotic. These two terms are not really interchangeable. Antimicrobial is a very broad term. It means anything that kills or inhibits a microorganism. Anti against microbe, right? So that term includes antibiotics, which are natural. They're made by an organism, as opposed to some synthetic drugs that we use, which aren't made by microorganisms. They may be extracted from plants. They may be chemically made in a lab. Okay? So a lot of times people misuse the term antibiotics. And is actually just an antimicrobial that's probably synthetic or from another source other than a microorganism. But same action, right? And that's what most of us care about, is that antimicrobials and antibiotics both are going to either kill or inhibit microorganisms. So why are these guys fighting? Like, why does this penicillin want to kill this bacteria growing on this petri dish? Resources, right? They're fighting over food and space. Same things we fight over, right? And they're using chemical warfare to do it, right? Y'all, this is chemical warfare. We just stole their technology, right? We stole penicillin from them and used it on our own, right? That's what we have done for a lot of the antibiotics, right? It's stolen technology. No, it's killing the bacteria. Penicillin will kill the bacteria, right? It will kill the bacteria. And that's why you see here, near the penicillium, where the penicillin is in here, this Staph aureus, this gram-positive bacteria, is actually killed. There's no growth here. Just like we saw those zones of inhibition, those no-growth zones on our plates because of the antibiotics, it killed that bacteria. Now, does it always work? No, what did we see today? We saw some resistance, right? We saw that penicillin had no effect on E. coli, right? E. coli, like, laughed at that stuff. You know, you were probably like, well, was there any penicillin in that disc, right? Did we put an old disc on the plate? No, it comes out that way every semester. It's because of the differences between these organisms. And that's why sometimes one antibiotic will work and one will not. It depends on what bacteria you're dealing with, right? And sometimes when they try antibiotic after antibiotic, that's when they finally do an antibiotic sensitivity test. They're like, well, what we're giving you isn't working. Let's figure out what bacteria you have and what antibiotics are going to work, right? But a lot of times they'll just throw out a broad spectrum antibiotic, and that means that it takes care of a broad range of bacteria, right? The problem with that is, do we have good bacteria in our gut and on our skin? Yeah, are they affected by the antibiotics? You betcha, right? And that could be sometimes more damaging than helpful, right? Especially when it comes to disrupting the microflora in your intestines. Leaves you more predisposed to other infections because they help you out by being there. So some other terminology related to how antimicrobials and antibiotics work, especially as it relates to bacteria. Some of the drugs are classified as bacteriostatic, which means they don't actually kill. They just slow down the growth of the bacteria. That gives your immune system a much better fighting chance at eliminating the bacteria. Bactericidal, on the other hand, actually kills the bacteria, those drugs. Bacteriostatic are helpful, right, especially under certain circumstances. But if you have a patient that has a compromised immune system, are you going to give them a bacterial static drug? No, because you're going to slow down that bacteria, but you're not going to eliminate it. And that person's immune system can't do it. It's compromised, right? So, HIV patients, you're not going to give them a bacterial static drug. You need to give them something that's bactericidal. You need the drug to take care of the bacteria for them. Huh? It depends on the infection and what's going on. There's lots of factors that would determine which drug you would use over one. But that's one of the main things that you would consider too. Is it going to actually kill, or is it just going to slow it down? Right? So some of the drugs that we looked at, chloramphenicol and tetracycline, are bacterial static. They just slow down the bacteria. Where beta-lactam drugs, which penicillin and amoxicillin, which is a modified penicillin, fall into that class. And you guys may have noticed that ampicillin did kill the E. coli. And that's, again, because penicillin has been modified into a lot of different psyllins. Ampicillin is an example. Amoxicillin is actually... it's a modified penicillin and a drug. No, that's augmentin. Amoxicillin is a modified penicillin. But they've been chemically modified so that they're more effective against gram-negatives than plain old penicillin. Because we saw plain old penicillin, E. coli, gram-negative, laughed at. It was like, you can't get me. And it has to do with how these drugs work. And that's one of the things that we're going to look at next. And the good news for you guys is I don't want you guys memorizing which drugs are what or how these drugs work, because things change over time. It's understanding the general ways in which these work, right? We're not going to memorize for specific ones, although most people know about penicillin. It's the most widely used and talked about since Alexander Fleming discovered it way back when, right? And purified it from the fungus. So how do they work? And they work differently depending on the antibiotic. So, for instance, chloramphenicol interferes with going from RNA to protein. So it interferes with ribosomes, right? With their ability to translate the information in RNA into proteins. And that's a very vital mechanism for a cell, right? So interfering with that can be detrimental. Remember, chloramphenicol, as I said, is bacterial static. This is definitely going to slow you down if you can't make proteins, right? And it's effective against both gram-positives and gram-negatives, right? So that would be a broad-spectrum antibiotic. So you should have seen that it was effective against both our E. coli and our staph today. Ciprofloxacin, commonly abbreviated Cipro, interferes with DNA replication. So the cells can't copy their DNA to make new cells. So it is effective against gram-positives and gram-negatives. Where penicillin, straight up plain old penicillin, is only effective against gram-positives. Our gram-positive we were using was Staphylococcus epididymis, actually. And this one has a different target. And the target is what really makes that difference as far as gram-positive versus gram-negative. Because it's interfering with cell wall synthesis. And remember, who's got that really thick layer of peptidoglycan? Gram-positive. And gram-negatives have a thin layer, but they also have an additional layer that's really protective for them. What's that additional layer that gram-negatives have?
The outer layer.
The outer membrane. And that outer membrane really stops a lot of drugs from getting in, right? Even ones that are dealing with the cell wall is synthesized inside the cell and then transported outside the cell. So all these processes are happening inside the cell. So where do the drugs need to get? Inside the cell. So if it can't get in, it can't interfere with these processes. And that's one of the major protections that gram-negatives have, is that outer membrane. It actually stops a lot of antibiotics from even getting in. Right? So it's naturally resistant by having that outer membrane. So as we see, right, our gram-positive is very thick peptidoligand layer, gram-negative is thin, but really what's really important here is this outer membrane. It really helps protect them. So in peptidoligand, the strands that are held together are cross-linked by the amino acids, and that's when this drug goes in, it interferes with that mechanism that makes those cross bridges that connects the sugar layers, and therefore really detrimental to gram-positives. So some more of the drugs. So streptomycin, this one, again, interfering with ribosome, right? So it's interfering with protein synthesis. And this one is effective against E. coli. But most gram-positives are not affected by it. Tetracycline, again, interfering with the ribosome with protein synthesis. And then ampicillin, as I said, is a modified penicillin. So it actually can get into gram-negatives and interfere with their peptidoglycan synthesis. So it is effective against E. coli as well as against staph. We didn't bother testing it against staph because, did you guys notice how huge the penicillin zone of inhibition was? Like that plate, I could only put one other antibiotic on it because otherwise the zones just run into each other and you can't measure it. This zone would be huge too because it acts, it's basically penicillin. So it acts the same. It's very damaging to gram-positives. So we didn't bother doing a plate for that one. But wanted to show definitely the effect that this modified penicillin has on E. coli, right? That this modified penicillin can affect gram-negatives like E. coli. And we're going to use this antibiotic later in our transformation experiment towards the end of the semester, where we're going to make E. coli ampicillin resistant, right? So it will actually grow in the presence of ampicillin. But we already know right now, natural good old E. coli, right, non-transformed, is killed by ampicillin. We've seen that in our experiment that we set up. Sulfur drugs, which I think we have, I can't remember we got a new one in that's in your lab manual, but we didn't mess with it this semester. But sulfur drugs, when you hear people talking about sulfur drugs, and again, these are drugs, these are not antibiotics, but they are antimicrobial. They will affect microbial growth. These particular drugs, sulfonamides, interfere with folic acid synthesis, which a lot of the bacteria in your gut do for you. They make folic acid for you. We don't have the enzymatic pathway to make folic acid. We have to either ingest it in our diet or the microbes in our gut have to make it for us. And they found that it's really important for what, women? Anyone know when you're supposed to take a lot of folic acid? When you're pregnant. Apparently it's very important for the developing fetus. So they want you to increase your levels by taking a vitamin. So this actually interferes with bacteria's ability to do this. And folic acid is needed for vitamin B9 synthesis. So it's a very important molecule. And it's important for us, but it's also important for the bacteria. And so stopping them from being able to do this will kill them. And that's how sulfur drugs work. It won't affect us, right? We don't have that pathway. We can't make folic acid. We have to either ingest it or our microbes are going to make it for us. So how many of you have got this in your medicine cabinet or first aid kit at home? Good old triple antibiotic ointment, also sometimes referred to as Neosporin. So how many antibiotics are in it? 3. So it's a 1, 2, 3 punch, right? And guess what? They all act differently, and that's the whole purpose, right? Is that, we're not giving you 3 drugs that all work the same. We're giving you 3 drugs that each one of them are going to hit a different target. So what are these drugs and what do they do? So polymyxin B interferes with and disrupts plasma membranes. And the bad news with this one is that it can be toxic for us too, which is why, in a moment, it says what it says as far as triple antibiotic ointment. Next one is bacitracin, which is another one, right, that interferes with cell wall synthesis, but it's another one you don't want to take internally because it can be toxic. The next one is neomycin, hence the name Neosporin, right, the brand name. And this one interferes with protein synthesis. So we're hitting 3 different targets here, right? We're hitting membranes, we're hitting cell wall synthesis, and protein synthesis. Bacteria are unlikely to develop resistance against all 3 of these antibiotics, right? They might be resistant to one, but the other 2 would then still be effective. So that's another reason why not just do 1 antibiotic, but do 3, right? The chances of whatever you're dealing with of being resistant is much lower, right, than if we were just dealing with 1. So yeah, external use only, right? You don't want to take this internally, right? You got to call poison control if you do. And it's because the polymyxin B and the bacitracin are toxic for us if you were to take them internally. It also says, let's see, for minor cuts and scrapes and burns. You don't want to cover your whole body in this stuff, right? Because then you would have the potential of absorbing too much into your body, right? And it would be not all that different from swallowing it, okay? So it's for small areas. And you're going to clean the area. And nowadays, you know, some people in the past would use hydrogen peroxide. We found out that that actually causes quite a bit of damage to even your own cells. So you're better off just soap and water and a little bit of triple antibiotic ointment. But you do want to try and clean first any debris, any dirt, or anything like that. And you're going to put a small amount on the tip of your finger. And actually, I even suggest putting it on a Q-tip to keep anything that's on your finger from getting on your ointment, right? Because then you could contaminate your ointment potentially. And you want to keep it clean. Stop use and consult doctor if condition persists or gets worse. Systems, right? Obviously, if it's not getting better, right, these antibiotics aren't taking care of it. You may need something else, right? This isn't going to take care of everything, but it's going to take care of most things that we encounter, right, when we have a cut, scrape, or small burn, right? We're not talking about a pot of water on your entire arm. That's an emergency room visit, no doubt, right? So, and at least one, the antimicrobials is toxic to non-skin cells, right, in the concentrations found in the ointment. So how does this resistance problem happen? Well, as we said, what has to happen? The cells have to take up the antibiotic, right? And it's going to interfere with some mechanism inside. In the case of ribosomes, right, and protein synthesis, the drug usually binds to the ribosome and interferes with its functionality. Or it binds to the enzymes needed for peptidoglycan synthesis and interferes with their ability. So how do they become resistant? Well, some of them decrease the uptake, and definitely with gram-negatives, one of the problems is that it can't get past that outer membrane, right? It can't get in to begin with, so therefore it can't be effective. Another problem is some drugs are taken in, but the cell recognizes somehow that it's bad and kicks it back out, right? He's like bailing the boat. No, 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 you're bad stuff. Out, 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 out. You can't sit, right? Or the antibiotic or my antimicrobial comes in, and that bacteria has enzymes that destroy it. And this is very common, unfortunately, with penicillin, like drugs. There's a lot of bacteria out there that have enzymes that destroy penicillin and penicillin-like drugs. Penicillin, its general structure has what's called a beta-lactam ring here. And so there are enzymes out there called beta-lactanases that literally cut and break this ring and therefore deactivate penicillin, stops it from binding to the enzymes involved in cell wall synthesis. This portion of penicillin is what's changed or chemically modified depending on what derivative or modified penicillin, right? So this is what's different for amoxicillin, ampicillin, all these other cillins that are out there. The other problem is if the drug is taken in, but notice the target has changed, right? So the drug can go in and bind to the target here, but it can't recognize it anymore. So if some of them have, their ribosomes have changed so much that the drug can no longer bind to the ribosome and affect protein synthesis, right? So if they mutate, right, if they change such that now the drug can no longer bind to the target, it's not going to be effective. So, I don't remember why Peter put that in there, so that's just binding of amino acids. So how does selection work? So you have this population of bacteria, right? And you apply this selective pressure. And in the case of what we're talking about today, that would be an antibiotic in the environment. The ones that are sensitive, what's going to happen to them? They're sensitive to the antibiotic. They're going to be killed off. But if they're resistant, they're going to survive, right? And those cells are going to then be the predominant ones in this population. So now, right, sensitive guys are killed off by that selective pressure of the antibiotic, and you're left with the resistant, right? And then the resistance will dominate the environment. This phenotype, this ability, is encoded in their genetics usually, right? To make, say, an enzyme to destroy penicillin, or to make a different ribosome that tetracycline doesn't bind to anymore, or the ability to kick out particular antibiotics is coded all the way down in their DNA, in their genotype. What you can do with your DNA, that physical expression is known as your phenotype. So the phenotype is, say, making that enzyme that destroys penicillin. This is natural selection, right? Survival of the fittest. And this has definitely been shown with that selective pressure of antibiotics as it applies to Staphylococcus aureus. MRSA, or sometimes pronounced MRSA, stands for Methicillin-resistant Staphylococcus aureus. Methicillin is a penicillin derivative that was commonly used for Staph aureus infections. So much so that some resistant strains came about, and now they predominate. The bad news is, right, so, okay, we can't use methicillin anymore, so we're going to use something else, right? The next drug starts with a V. Anyone know what the next drug that we now have staph that's resistant to? Yeah, vancomycin. Vancomycin. So we have now vancomycin-resistant strains of Staphylococcus aureus. This battle will continue. The bad news is we may lose in the end, because we'll run out of antibiotics, right? And this is why drug companies, right, work so hard to try and get new drugs, because resistance happens, right? Organisms are constantly changing, right? There is the potential that antibiotics will no longer be useful. So if you make a medicine for all 10 different shapes, then... Yeah, but how do you make a medicine? That's not as easy as baking a cake. Right? You know, there are actually endless possibilities. Yeah. Not necessarily its shape, but, you know, the molecules that make up and do the functionality. But, yeah. I mean, I guess, you know, if they could figure out down to the molecular level what part of the ribosome is most consistent and never changes, then we can make a drug against it. But never changing is never going to happen. Right? Things are always constantly changing. Right? There's going to be something that's going to change, and that drug isn't going to work anymore. Yes. This is why they're prescribing less and less antibiotics, and also why they're being more cautious. And this is why we have to as educators, right, need to help the general populace. People go to the doctor with the flu and they think that if you get antibiotics, you're going to get cured. It's not going to happen, y'all. You have a viral infection. They can confirm that even now, right? And the sad news is they do have some antivirals out there, which is good. But for some infections, they're going to tell you go home and rest, right? Drink, rest, eat. Pray your immune system does its job. We're going to give you some supportive care, but your immune system has got to save your butt, right? And so there, sometimes, especially with viruses, yeah, you are really just dependent on your immune system saving your butt. With some bacteria, though, we can use antibiotics. The problem is people misusing them, not taking them for the full course, right? Oh, I feel better. Well, that doesn't mean you've killed all the bacteria. And if you stop taking it, there's more likely that that the resistant ones, if there are any, are going to predominate that population. And then you've got to do a different antibiotic, and then, like I said, we're going to eventually run out. When it comes to Mycobacteria tuberculosis, that causes tuberculosis, that bacteria is evil. And it's developed a lot of resistance. There's strains out there that are extremely drug resistant. And the problem with that, too, is patient compliance. Is that infection, the architecture of it and everything, some of those drug treatments need to be 6 and 9 months long. And patients stop taking their medication, or they stop being able to afford it, or any other obstacle. And then that has really led to the resistance that we see, is really the non-compliance. Yeah. Because it's such long term. It's expensive. But they have special programs, they try to monitor and keep up with patients and make sure that they do what they're supposed to do. And it's so hard, too. I remember when my son had some ear infections before we figured out that it was allergy related, and the fact that, little kids' ear canals don't drain well. He had to have surgery and part of his adenoid removed. And I had a point here. Yeah. I lost it. It's because my head is killing me today. Yeah, it's all right. It's all good. But you guys get the idea, right? You get the idea how important this is.
Just real quick, in your lecture class 2 years ago, you mentioned if someone spit that annoys you because there's spores.
It's not spores, it's bacteria, right? You have, especially if you have a cold, right, and you're producing excess mucus so much that you don't really want to swallow it, but you really should, and you feel the need to spit it out of your body, you're exposing everyone else to your bacteria and viruses. By spitting it out of your body, it becomes part of the environment, right? It's airborne and, on whatever it is you spit on. Yeah, become aerosolized, but yeah, we won't get too germophobic right now. Okay, so moving on. So, for all the antibiotics out there, there are some microorganisms that are resistant. It's just going to happen, right? It's just going to happen. So chloramphenicol, right? Some bacteria don't take it up, or they can inactivate the drug. For ciprofloxacin, again, poor uptake or alter of the target. In this case, the enzymes that do the DNA replication are no longer affected by this antibiotic. For penicillin, poor uptake is definitely the case with gram-negatives or straight-up penicillin. Production of beta-lactanases, as I said, those enzymes that actually break and destroy penicillin. And alter of the target, where the enzymes that penicillin actually binds to have changed. So for all these guys, right, there are ones that are resistant to streptomycin because, again, poor uptake. Production of an enzyme that's actually encoded on a plasmid, that's really bad news because plasmids can be easily shared from bacteria to bacteria. That genetic information on how to do that. So the way this method works, right, we confluently inoculate about the same concentration of bacteria, which we do either by eye if you have a good standard or using a spectrophotometer. And then we place discs impregnated with a specific concentration of the antibiotic on them. And so they're all coded, right, with letters and numbers so that you know which antibiotic is what. For you guys, in case our antibiotic disc came detached, right, we even wrote the letter above the lines. And I drew these lines the very next day when they were the most accurate. So you'll notice that maybe some of those might have encroached in growth-wise, or the zones might even have gotten bigger as time went on. But that line was when they were accurate, and that's why they were drawn for you guys. And we definitely saw some complete resistance, right, where it grew right up to it. So we refer to this as the zone of inhibition, the area where the growth of the organism, of course, is inhibited by the antimicrobial agent. So minimum inhibitory concentration, this is the lowest concentration of a specific bacterial static antimicrobial drug to prevent the growth of a given bacteria. In vitro, what does that mean? In the petri dishes, right? Not in the body. In the body would be in vivo. And body factors do affect, right? The rate of metabolism by the liver, the rate of removal by the kidney, and things like that. And that's what I wanted to say about my son. So many freaking antibiotics, right? So, like, by the time you get to, like, the 4th round, being a mom, you're like, I don't care how much the antibiotic costs that you only have to give once a day, but I want that one. Right? Because there's 2, 3, 4 times a day. But that has to do with the half-life and the breakdown rate of that antibiotic within your body, right? And unfortunately, the ones that have a lower a longer half-life, right, a slower decay rate, tend to cost more money, right? But when you're, by your 4th round, you're like, I don't care. Because it's really important at keeping those levels steady, right? When you have an antibiotic you give twice a day, you really want to try and give every 12 hours because you want those levels to stay steady. Because if they drop down, then the bacteria grow back, and it's going to take a longer dosing of the antibiotics to really eliminate those bacteria. So those are other important factors that sometimes people don't really understand when they're given antibiotics too. How important it is to take it in a timely manner. So I knew it would come back to me. So minimum bacterial cytal concentration, that's toxicity. Again, we want to know what's the lowest concentration, right? We don't want to give you too much because we don't want to worry about possible interference with our own cells or toxicity levels. And that's really important, right? We don't want to be, we want to be lethal to the bacteria, right? So, what can we give that's going to be lethal to them? Right? So sometimes that's even measured, right, as the mean lethal dose. Toxicity matters, right? So especially for those topical antibiotics, you don't want to be taking the stuff internally because it's toxic, right? We want to make sure that we're targeting them and not us. And especially with cell membranes, right? Ours aren't really all that different from theirs, right? So those types of drugs are usually not taking internally because we would cause too much destruction to our own cells. A little bit on your skin doesn't really bother you, too, plus the outer layer of your skin for the most part is dead anyways, right? You slough it off every 48 hours, which is why eventually the color will go away on my hands as the skin cells are sloughed off. So this is a little bit different test. This is an E-test, and it uses strips instead of the discs. And on the strips is a concentration gradient. And it is indicated on the strip itself. So where it bisects, where, it's after this point, notice it's not inhibiting growth anymore. This is your minimum inhibitory concentration, right? And see where it's much more concentrated, it kills more bacteria, right? The zone is much wider. So, minimum inhibitory concentration, regardless of whether the antimicrobial is toxic, which is bactericidal, or merely inhibitory bacteriostatic. Some people get real tricky and real specific on the terminology. We're not going to get anal about it. Okay. So how does the antibiotic get into the media? Simple diffusion, right? But the important thing to realize here too is it doesn't only go out, and of course as it spreads out its concentration is going to decrease, right? So even though we're working with a single disc as opposed to that strip with the gradient, we're still going to have a gradient, right? The greatest concentration of the antibiotic is going to be closer to the disc, and then it's going to lessen as it diffuses out. So we really are measuring the minimum inhibitory concentration in our test. We're just not setting a value to it like they do in the e-test. But the other important thing to note here in this diagram is not only spreading out, but it also spreads down into the agar. And this applies to one of our questions as to the depth of those plates. So I poured those plates for you guys, but I poured them from tubes that contained 25 milliliters of the media. And the reason for that, it would give us, if you were to measure the thickness of those plates, they're about 4 millimeters, right? We want to standardize that for this test because not only does it spread out, it also spreads down. So what if we screwed up? Right? And I actually saved a plate. It's in the fridge. I'll take it out for you guys later. It's too thin. So what's going to happen if I had used that plate? What's going to happen to our zones? They're going to be larger. So are they going to be accurate though? No, right? Because they can't spread as far down, they're going to spread out wider, right? So they're going to be larger zones and they're going to be inaccurate. So the thickness is very important, right? So it was a measured amount of media that I poured into each one of those plates, pre-measured, to make sure we got the right thickness. It was also a special type of media called Mueller-Hinton 2. So again, we standardize what food they're getting, the thickness of the media, because diffusion is how this test works. The other thing that's important, so of course it says you'll be pouring them, but I did that for you. I did change that. So, you can mathematically calculate it. Peter loves the math, right? But here it is for you. And as I said, you need about 25 milliliters for our standard Petri dish size to get 4 millimeters thick. So the plates are supposed to use within a specific time after the preparation and should be free of visible moisture. Why would you be worried about moisture? I actually had to throw out some plates because they were too moist. Remember, we're dealing with what? Diffusion. And so if they're too moist, how is that going to affect the diffusion? Is it going to make it more or less? It's going to make it more. Actually, a wet paper towel, if you drop some dye on it, right, the water absorbs the dye and it'll spread. Where a dry paper towel, it'll pretty much stay where you put it. So it would increase the diffusion, though. It would screw up your diffusion rate. And again, it would make your results inaccurate. And if they're old, then they're going to be dry and your diffusion's not going to be enough, right? And your zones are going to be too small. So you need it just right. You need Goldilocks, right? Because moisture does affect diffusion. In clinical applications, you would base it on the McFarland standard, and you'd use it within 30 minutes. So about how many bacteria do they say in your lab manual 0.5 McFarland is supposed to be? I think 1.5 times 10 to the 8th. Is that right? Something like that. No one wrote it down? It's on my next slide. Aha! Look, I remembered right. Wow, that's amazing today with this headache. 1.5 times 10 to the 8th. That's 150 million cells. Wow, that's a lot. So what happens, like, for E. coli after 30 minutes? So there's still going to be only about 1.5 times 10 to the 8th cells? No, there's going to be what? Almost double that, right? That's why I had to make it fresh right then, right? So that our cell density is within the range we need. So we use two organisms, and the reason why we use these two different organisms is so that we had a representative gram-negative and a representative gram-positive. Because as we saw, especially for penicillin, that cell wall structure really makes a difference. So they gave you this example. Oh, wait a minute, I'm getting ahead of myself. So, of course, why do we see these differences between gram-positive and gram-negative? Because they are structurally different. Do you have a question? Oh, yeah, might be. Yeah.
It was really loud. We were like,
Yeah. Glad we're in here, right? We'll just stay here. Okay. So remember, these differences are really important, especially as it relates to penicillin that affects cell wall synthesis. But again, for gram-negatives, having this outer membrane really sometimes really helps them from the antibiotics just even getting in. So this is the one I was thinking of. So we have a staph species, and they tested these two antibiotics, penicillin and chloramphenicol. And when they measured the zones, they came out the exact same diameter. But it doesn't really matter, like, one compared to the other, right? Because how do we read this test? You have to look at your chart, right? And the good news is, of course, you're not going to memorize this chart. I'm going to give it to you on your quiz next week, right, y'all? Right? You just have to know how to read and interpret this information. So it says for penicillin, in order for it to be susceptible or sensitive, that zone has to be 25 millimeters or larger. Is it in this example? It's only 25. So this staph is resistant to penicillin. Would you give that to your patient? No. So the chloramphenicol, though, that one has to be 18 or greater. So is it susceptible to chloramphenicol? Yeah, because it's 25, which is greater than 18. You have to take each antibiotic individually, right, and compare it to your chart. Don't go, oh, well, you know, this one's bigger than the other one, so therefore this antibiotic is better than the other. No, no, no. It doesn't work that way, right? You got to go by your interpretation chart to determine susceptibility or resistance to that specific antibiotic for that specific organism you're testing. Right.
Are the concentrations the same for those two antibiotics?
The concentrations were probably different. You know, and that's going to depend on the antibiotic. So again, remember, this test works on diffusion. So sometimes it's referred to as the dish diffusion test as well. Because you're using discs and the antibiotics are diffusing. So after incubation, does the antibiotic extend into the agar beyond the zone of inhibition? Do you think the antibiotic keeps spreading out? Yeah. But why don't you see bigger zones? What happens to the concentration of the antibiotic as it spreads out? It's weaker, right? Where you see that stop, that edge, is your minimum inhibitory concentration, right? The minimum amount of the antibiotic where you're still killing the bacteria. That's rain or is that a cart? I'm like, it sounds like a cart to me. All right. Okay. So, you know, sometimes you could do dilutions, individual dilutions, right? This is the concentration of the antibiotic. And you can see, of course, at zero concentration at the control that doesn't kill the bacteria, it's cloudy. At 1 microgram per milliliter, it's not killing the bacteria. But 2, at the lowest concentration of 2 micrograms, right, for this particular example, they're killing the bacteria off. So that would be your minimum inhibitory concentration. Again, we don't know the concentration in the disc diffusion test or the Kirby-Bauer test, But we're not really asking that question with this test. We want to know, is that particular antibiotic effective? Is the bacteria sensitive or resistant to that particular antibiotic under these standard set of conditions? So, right, we have our set table of susceptibilities and resistance and intermediate. These are some older values. I bet your values were very similar to this. So, of course, for streptomycin, right, it has to be 15 or greater. So, for E. coli, it was sensitive. Where's my S? There we go. For Staph aureus, though, in this particular run, right, it's just barely sensitive, right? And same thing for Pseudomonas. Or sometimes I do those plates. This time I didn't feel like it. This is another gram-negative, but it's resistant to a lot of antibiotics. And that's one of the reasons why I usually do those plates for you guys, so you can see that only like 1 or 2 end up working. So for tetracycline, remember you got to look at your chart, so that's 19 or greater. So the first 2, E. coli and Staph, are sensitive, but as I said, look, the Pseudomonas is resistant to tetracycline. And since it's a gram-negative, you expect the same result for penicillin as we do with E. coli, right? 100% resistant, no zone to measure, grows right up to the disc. Unlike Staph, the gram-positives, which penicillin is very effective against, you get that nice large zone. Penicillin has to be 29 or greater, and definitely the case here. So we have 2 naturally resistant gram-negatives and then a sensitive gram-positive. For chloramphenicol, it's got to be 18 or greater. So again, these 2 guys, the E. coli and Staph, are sensitive, but again, we have the resistance here for the Pseudomonas. So this is all the data from all the plates one semester that we did. And I added the black bars that have the sensitivity. So it has to be at this bar or above to be sensitive. And again, you notice Pseudomonas here in green. We only had measurable zones for ciprofloxacin, which it was sensitive to. Notice it's above the black bar. It was just under for streptomycin in that run. All the rest, we didn't even have measurable zones. They were 0. That's why you have a blank space here. Blank. Blank, blank, blank. Didn't even, it just laughed at it, right? And then for E. coli, again, we see that 0 here, right? E. coli would be right here for penicillin. There's no bar at all because it's 100% resistant. For our Staph, it was pretty much sensitive to everything, right? Staph, this pretty blue bar, is above all of the sensitive bars for all of the antibiotics that we tested. When E. coli was sensitive, again, to most of the things as well, right? So streptomycin, tetracycline, chloramphenicol, ciprochloramphenicol, but definitely not penicillin, naturally resistant. And it is sensitive to ampicillin. E. coli is sensitive to, I can't even say this big a long name. But it is resistant to vancomycin, E. coli. Gram-negatives are. So Peter Hickman, one of the instructors that teaches this course as well, had a really scary bout with some Staph aureus after a poison ivy incident, right? So he had poison ivy and then the skin got infected with Staph aureus. He's allergic to penicillin. I couldn't give him to penicillin. And actually had this golden discharge, and Staph aureus, aureus actually means golden in Latin, and it can grow kind of yellowish sometimes. So one of the docs recognized that, said it's probably a staph infection. And they had to, of course, put him on a different antibiotic than penicillin because he's allergic to it anyways. Yeah, double whammy. So our scientific method for antimicrobial susceptibility testing. That brings us to enumeration of bacteria. So there's different ways you can count bacteria. You could do a direct cell count, which with bacteria you definitely probably wouldn't want to do this method because it's not easy. And as you know, bacteria are teeny tiny. But this method uses a special slide that has a special fixed chamber in it that has a set volume when you fill it up. And it has a grid so that under the microscope you can count. You can also use a dye that will dye the dead cells and not the living cells. But usually we're concerned about living cells, right? So again, not the best ideal method for bacteria. This method is used a lot for eukaryotic cells. When I was doing research and I had to figure out my cell suspensions, I would use this hemocytometer chamber slide in the stain. And then I can mathematically calculate because you know the volume. Indirect counts, like measuring biomass or measuring biological products, sometimes can be helpful if you know the production rate and the possible starting estimated amounts. We did serial dilution using the standard plate count method. The drawback with this is, of course, we have to wait till tomorrow to get our results, which is why we're always waiting on our water to be tested, right? Because they're doing this. They're trying to find out how much of the bad bacteria are in that water sample or those water samples they took. So we dilute in series because that's much easier, and we mathematically calculate the total or the dilution factor for each tube in our dilution series. Another way is utilizing a spectrophotometer, which we did do today, so we will be able to correlate that to our results, and also gave us a general idea based on what we know for the McFarland standard of turbidity about how many cells we might have, right? So we're thinking we might be around McFarland standard, which is 1.5 times 10 to the 8th, right? So like close to 1 billion cells, right? And it wasn't very cloudy, right? That's a lot of bacteria. So the way the spectrophotometer works, right, it measures the amount of light that passes through or doesn't pass through. This is set for transmission. So it's measuring the amount of light that passes through. And then you could directly correlate that to the cell count numbers. So no more of these bulb things, right? And having to worry about meniscus in large volumes, right? We didn't mess with that today. That's pretty much going by the wayside unless a particular protocol calls for that. We tend to use much smaller sample size and utilize the micropipettors we use today. So pipette men and how we read them, right? So this would be 1.5 for the 20, the P20, which has a maximum volume of 20 microliters. In this case, this would be 15 microliters. For the P200, these are nice. Pretty much what it says is what it is, right? This says 20, this says 200, it is 20 and 200. The P1000 goes to 1000, that's the highest. And so that red value at the top denotes, you would add one more zero here, and it'd be 1,000, which would be an entire mil of solution. Where here, when you set it at 20, you have to add the additional zero. It would actually be 200 microliters. Right? We put disposable plastic tips on, right, to suck up the solution, and it controls the volume for us by the setting on the knobs. This takes a little practice, right? And later on we'll do another experiment where you guys will actually get to use these types of instruments.
So we're not going to do lab 7 project today, or?
We already did it. We did that during the during the midterm exam. Yeah. We're not doing anything more but talking about dilution today, unfortunately. So how do we count? So I just left this in here. Those of you guys that are struggling with good old Common Core math, like I talked about in my lecture class, one of the things that Peter mentions in his history of counting in here that he put together, which is really cool, is this use of, I'm trying to get to it. Zeros are placeholders, right? Place value holders, that's what a zero is. right? I was like, oh, I never thought of it that way. That makes so much sense. Especially helps when you're trying to teach place value to a little kid, right? And that's all they are. It doesn't stand for anything, right? It's 0. It's nothing, right? But when you have a 1 and a 0, it's 10, right? Because now we've gone to the next place value, which is the 10s, as opposed to the 0 that's holding the place for 1s, right? So this goes over a little bit of place value and writing things in expanded form, right, which can be helpful. And math can be scary for people, so these actually are really good examples that I think do a pretty good job of explaining, although I'm not going to sit and go through them. But they're there for you. And then scientific notation, when we do our final calculation, we need to write that in scientific notation. So scientific notation is using exponents, right? So 10 written as an exponent. And you're always going to have one whole number and then your decimal. So in this case, right, we move it decimal over 1, and so that's 10 to the first power, which is basically just 10. For 200, right, we move our decimal 2 places, right? So it's the 10, right? Each one of these is 10s to the second power. And this is how we write each one of these, right, in exponential or scientific notation. So the important thing is a single whole number and then decimals. And usually 2 decimals, although this one just has 1. I don't know why. So the how we measure, not the system that we use for measurement, but the metric system, is actually based on place value in groups of 3, right, and groups of thousands. So so for instance, gram is the base value. Kilo refers to 1,000 more grams, and then when you go smaller, milli means 1,000 less of grams, right? And picograms is another power of 3, so now it's 1,000,000 and 1,000,000,000 times. And then what comes next? Trillion, right? Trillion times small. And I didn't femtogram. I've heard of picograms, but we can go to 15 zeros below. Yeah, it starts hurting my brain. Really small, right? Really small. So mathematical calculations, right? I sometimes refer to, and Peter doesn't like this, but to me it makes sense. Fractions are evil Siamese twins. They're stuck together. And I call them evil because if you don't do the same thing to both sides, they get mad. In other words, you get the problem wrong, right? Right? So with fractions, if you are going to multiply the top to get the number you want, you have to do the same thing to the bottom half, right? The denominator. The numerator and denominator, they're stuck together. You got to treat them the same, right? They're just like twins sometimes. You got to buy them both the same damn toy. Otherwise, they get upset. So there are different mathematical calculations, examples of, right, how to multiply and divide fractions. So when you divide, remember you multiply by the reciprocal, which means you flip it. Okay, and that's what this is denoting here. So m divided by n is the same thing as m times its reciprocal, which is 1 over n, right? If you take n and flip it. And of course, any number multiplied by 1 is that same number. So there's examples of each of the mathematical principles. If math is tough for you, y'all, I love math. Come sit with me during office hours or make appointments with me. I will go through all of this with you, step by step by step. I think math is cool because you always eventually get the right answer. We just got to, figure out what method are you going to use to get there and do it consistently and correctly. So why do we make serial dilution? So why did I do all that today? It's going to allow us to make extreme dilutions, right? I mean, I diluted, what was the final number? I can't even remember. I was diluting almost 1,000,000 times, right, from my original sample. If I was to do that into a large sample, it just wouldn't be impractical. If you were to take, say, one of our lab benches, right, and do a set volume of water there, we're talking about, 327 gallons it would take to fill up one of these lab benches, the volume that one of these lab benches makes up, right? That would be 1.6 tons, 1.36 tons. Wow. That's a lot, right? Really just not practical. So if we do it in series, though, right, we can very easily reach very high dilution rates with much smaller volumes, as you observed today. So, some terminology that's utilized when we talk about serial dilution. So, we start with an original culture, right? And we're going to dilute into individual tubes. When we do that, we need to take into account how much we transfer, what our diluent is, and what our final volume will be. Sometimes this is referred to as the whole amount, right? So your transferred amount and dilution amount, that's your whole amount. To figure out your dilution factor, a lot of times it's written as a fraction, right? And some of us feel the way I do about fractions. They're evil Siamese twins. They're not necessarily our best friends. So, right, so we have our individual tube, right, and that is our full dilution or our dilution. Then we need to know the total tube dilution, because once we do more than one tube, right, we have to take into account all the tubes after that. So we refer to that as total tube dilution or dilution factor. So sometimes you're like, oh, what are we talking about, right? So these are some terms we're going to have to learn and try and use, right, and have a working understanding of them. So in your book for dilution of an individual tube, they do the part that you transferred over the whole amount you end up with in that tube. So that in this example, the amount you transferred is 1 ml, plus the whole amount you end up with is the amount you transferred and the amount that was in the tube. So that's 1 plus 9. So you end up with 1 over 10. The milliliters cancel out and you end up with a fraction, 1 over 10, right? So you would say a 1 to 10 dilution. For some people, this is ambiguous and they get confused and they think, oh, I put 1 into 10. Not realizing that when you add the 1, it makes a whole volume of 10, right? So it's much easier for some people to say 10-fold dilutions instead of 1 to 10, right? It gets confusing. So I'll try to always say 10-fold, right? So multiples of 10. This fraction could also be written, right, as 10 to the minus 1. The minus indicates that it's the bottom. It would go in the bottom half if you were to write a proper fraction. We have been trying out past couple semesters, Peter and I, instead of using the fraction, flipping it and making it a whole number. But in order to do that, what has to be at the bottom of the fraction? What number? 1, right? So you have to make sure that all your amounts, right, end up being 1 on the bottom. That isn't always the case. Right? So look at this one. But you could make this so that it's a whole number, and guess what it comes out to be? 10. Right? So you just move your decimal place over, right? 1 and 1, and then it's 50 divided by 5 is what? 10. Notice I did the same thing to the top and the bottom. I moved the decimal place 1 plot. So here, right, you could just divide the top by the bottom, right? And you end up with what? 10. So this is what we used to do in the past, right? We would do in milliliters, 0.1 and 9.9. But I just changed it to microliters, right? So, same principle though. It doesn't really matter what the measurement is as long as the measurement is consistent. So if you do everything in milliliters, then all the milliliters cancel out. If you do everything in microliters, all the microliters cancel out. You just have to be consistent in your unit. So we need to know the individual tube dilutions. So we've already said what this one is, right? 0.1 into 9.9. Oh, we didn't do this one yet. We did 1 into 9, but this is basically the same. Almost, right? So 0.1 plus 9.9 gives you what? 10. So that's a 10-fold dilution again, right? Nope. We got a decimal point here. So let's write this one out. Right? So we need to know our total. We just figured that out to be 10, right? So then we've got to take our total and divide it by the amount that transferred. In this case, it's 0.1, right? That decimal matters. Right? So it is actually a 1 to 100 dilution. But again, we're flipping it, so it would be just 100. So 0.1 into 9.9, written out R method. 0.1 plus 9.9 is going to give you the 10 on the top, right? And then you have 0.1 at the bottom. You got to make this a 1. How could you do that? Move the decimal 1 place, right? You have to do the same thing at the top. So then that makes it 100. Right? Or in scientific notation, 1 times 10, or exponent times 10 to the second. So our first step, right, even though I did this in milliliters, I did the same thing. I did a 100-fold dilution in my first step. And then I did another 100-fold dilution, right? I did the same thing again. But then we changed it up here. And I don't remember what I did on mine, but in this example, it's a 1 into 9. Anyone want to guess what the dilution? 10. 10, because 1 plus 9 is 10, divided by 1 is still 10, right? That's our third dilution. So we did 100 times, and 10 times dilute. And then in this example, we can continue on with 10-fold dilutions. So as you can see, 100 times dilution, 10 times, 10 times, 10 times. So that's individual tube, right? Here's some more examples I want you guys to try, right? See if you can calculate and get the correct answer. So I'm going to zoom through that so you can't see them. In the student version of the PowerPoint, the answers aren't there. So see if you can do them. See if you can calculate individual tube dilution. Okay. And then we don't stop there, right? Because we're diluting as we go. So in truth, the dilution of this tube isn't just 10 times from our original. It's 100 times 100 times 10 times diluted from our original culture. And that's what we care about, right? That's what we want to know. What's this original one? What did we start with? So we have to multiply. And the good news is when you multiply exponents, what do you actually do to them? You add them. So if you write it in exponent form, it's really easy because then you just add. So we get our dilution factor for the tubes. So this one, the first one, of course, is what we made. But any tube after that is the combination of all the tubes before it, right? So this tube, too, is whatever dilution we use to make the tube plus the dilution before it. So, or multiplied by it, but in this case of exponents, we're adding, right? So that's where we get the 10 to the 4th for this tube. And then this one is another 10-fold dilution, so it's 10 to the 5th, 10 to the 6th, 10 to the 7th. So this one we are going to do. So if you have 10-fold dilutions, right, like the previous example, how do we calculate that? It's 10 times 10 times 10, right? Which is 1,000 times or 10 to the 3rd. Well, okay, say tube A, you have a 10-fold dilution. Tube B, you have 5, and tube C, you have 2. How do you calculate the dilution first for B? What do you do? So the dilution factor of A is 10 and B is 5. What do we do with those two numbers? We got to multiply. We're not dealing with exponents, right? So we can't add, so we got to multiply. So the dilution factor of the final dilution, the tube dilution or the dilution factor for tube B is 50, right? So now if we had to figure out what C is, what do we need to multiply? 50 times 2. Or we could start all over again, but why would you want to do that, right? You've already multiplied A plus B. You know that's 50. You could just times it by 2. But in essence, you are multiplying all the tubes before it, including that tube, to get its dilution factor. So here we say, can you do this another way? Of course, right? You just keep going, right? Once you figure out what B is, you can times by C. Once you figure out what one tube is, you just multiply it by the dilution factor for that individual tube. And you just keep going down the line. So in lab, remember we use the term colony forming units. And again, typically we do it in milliliters, right? So notice too that I wrote down 0.1 milliliters instead of 100 microliters as to what I put on the plate. So that's the most common volume. Even though I did it in microliters, I can convert it all back to milliliters, right? You can go either way as long as you do your conversion correctly. So again, 0.1 milliliters equals 100 microliters. I'm all right. And again, we don't usually do a whole mill onto a plate. So because of that, we have to take into account that volume. Because if I put a whole mill on that plate, am I going to have more bacteria or less than if I put a tenth of a mil or 100 microliters? Which is going to have more bacteria? The larger volume, right? If I put an entire mil on that plate, that's going to be a lot more bacteria than if I just put a tenth of a mil. So the fact that I only put a tenth of a mil or 100 microliters, I have to take that into consideration in my calculations, right? That smaller volume that I put on those plates. That makes sense to you guys? Okay. So we want to know the concentration, commonly referred to as the titer. And I'm not going to bore you guys with the figuring out and writing out the equation. This is the important equation. This is your CFUs, right? The number of cells you're counting on that plate, divided by the volume that we plated, by the dilution factor that we calculated for each tube in series. All right, so let's look at an example. So again, to be countable, we want it statistically between 30 and 300 colonies. Too many, I don't know about you, but I'm not counting over 100. Never mind 300, right? And 30 is, less than 30 is just too few to be statistically accurate. And if you watch that dilution video that I posted for you guys under Lab 7, that first one, it really helps explain, why you wouldn't count less than that. It would be really not very accurate. That's the range our book goes by, but, different protocols, different procedures may do different ranges like 20 to 200. So here's an example, right? So here's the individual tube dilutions. Here's the total tube or dilution factors for each tube. Here's the amount transferred onto the plates. And then this is the colony count for each plate. So on this plate, say there was 977. No one would actually count that. They would write too numerous to count. They would count this one of 120 and definitely 16. But which number would we actually use? The 120, because 16 is too small, right? It's less than 30. So that's our countable plate. So we take that value, 120. That's our CFU. We know that we put a tenth of a microliter or a tenth of a milliliter or 100 microliters onto that plate. We did a million-fold dilution, so that's 10 to what power? What's a million? How many zeros? 6. 6, right? So our formula, right? We need the number of colonies, the CFU. We got that 120. We know the volume we plated, right? And we know the dilution factor. It's 10 to the 6th. So then we just fill it in and calculate. And you can use calculators, right? Or you can, mathematically, this one's not so hard, right? You can very easily add up your exponents, multiply your numbers. And notice the units is CFU per milliliter. Like I said, typically we do it in milliliters. Which even though I put 100 microliters on there, I would convert it into 1/10 of a milliliter. In those practice problems, those 5 practice problems I have for you, I'm really testing your knowledge to make sure you know how to do dilution. So sometimes, right, it's not multiples of 10. So you're not going to be dealing with exponents. You will actually have to multiply. The numbers aren't that hard, y'all. You can do this. I have faith in you. So if we had 0.05 milliliters of a 10 to the 4th, which is like 10,000 dilution, and you counted 200 CFUs, how would we set this up? So what would we write first? We would write our CFUs, which is 200. We would divide by the volume, which is what? 0.05, and then we would multiply by the dilution factor, which is 1 times 10 to the 4th. Right? You would set it up like that. That's probably your most important step, and then actually calculating the math. Let's do one more example. Right? So what comes first? So if the total volume of the original, this is different. This is one step further. So this is the concentration, right? Per milliliter. What if your original one, and I didn't measure mine today, but I could figure it out because there's a set volume in those tubes, so I'll figure that out. So we can do this for ours too. I think they're about, 6 milliliters or something like that. But say we started with a 200 milliliter flask, right? And we got this concentration. All we do is multiply the milliliters by the concentration, and the milliliters are going to cancel out. And it'll give us around the number of cells, or CFUs, that were in that original culture. So, right, our titer, this concentration, times the volume of our original culture is going to give us a number of CFUs. So in this case, 8 billion cells were in that original 200 milliliter flask. About. The book, of course, sets it up a little bit differently. We have it there, right? But sometimes when I go over this with you guys, I just confuse you. So I'm going to skip it. So I didn't do this setup, right? I did something a little bit differently today. But I think that's it. Oh, here's my setup from today. So this is what I actually did today. This is, some guesstimates, right, of what we may get. So we'll find out next week what we actually got. So notice I did it in milliliters. So again, you can go between milliliters and microliters. I did it in microliters. I can't get the two straight today. So this is just showing you out conversions and how it's the same. And then just another calculation. So again, how do we set this up? If maybe next week we get 125 CFUs on our plate labeled 5 times 10 to the 5th, and I put 100 microliters, which is the same as 1/10 of a mil. What do we write first? 125 divided by 0.1 milliliters multiplied by the dilution factor, which is 5 times 10 to the 5th, right? And so then we get possibly 6.25 times 10 to the 8th if we ended up with 125 colonies, right? So we would expect maybe this number, and that's not quite as much as what we expected, but pretty darn close. So we'll see what we actually get this semester, right? And I actually did staff, so just change it up a little bit, see what we got, right? So, And we have our OD reading we'll be able to compare it to, and I'll also figure out what the volume of our media that we started out with was. So, our scientific method, why would we want to do this? How do we do it? And what does it tell us? So the math is the tough stuff, right? So as I said, I got those 5 example problems. I know we're like at time for some of you guys, but if your brain isn't completely dead, try and do like 1 problem before you run off, right? See if you remember. If not, remember, as I said, come see me during office hours or make appointments. You can even call me on the darn phone, y'all. You have my number. I will talk to you, okay? So if you're struggling with this, right, rewatch this lecture. Listen to me going through those example problems, right? But we do have a quiz on this next week. And Kirby-Bauer. Kirby-Bauer is a walk in the park, right? It's going to be just like the homework problem that you had. I'm going to tell you the measurements. I'm going to give you the chart. You tell me whether it's susceptible or resistant and what antibiotic you would give your patient, right? And what's important about that test, right? All based on the questions. Yeah, woo. Yes. For you and me both.